And now for our final talk. Hang on, we'll get some AV stuff happening here. There we go. Yeah, we're getting better at this. Final talk was by Curtis. Curtis comes to us from the US, in the Boston area where he runs a facility there. But surprisingly, this is an expat. So originally from Canada, University of Ottawa, chemistry degree, I believe. And then he wandered over the border and is running some pretty cool stuff there. And he's here to talk to us about his adventures in automation. Come on up, Curtis.
All right. Well, I want to start by thanking the LRIG Canada organizers for inviting me to come today to talk about this talk at this event. Uh, I've been working at the Institute for Protein Innovation for the last 3 years, and one of the things I've been working on is coming up with ways that we can train emerging scientists and engineers for the automated lab. And we've been training some principal scientists along the way too. They usually need the most training. For those of you who haven't heard about IPI, we're a nonprofit research institute. We like to think of ourselves as a bit of a hybrid. We've got the freedom to pursue our research interests, just like an academic lab, but we also have access to the high throughput and scalable technologies that are traditionally found in a more industrial environment. And this allows us to really integrate academic freedom and industrial scalability to take on projects for impact, not just the profit motive that you traditionally see in the industry. The mission of the Institute is to advance protein science and accelerate research and improve human health. And we do that through 4 different pillars. We have our platform. So we have an antibody discovery platform, and we have an automation platform that underpins the entire Institute. We have our products, so the antibodies that come out of the platform, but we also make other tools available. So recently we deposited a paper into bioRxiv where we did a codon optimization study in mammalian cells. And as part of that work, we open sourced our protein production vector. And so this is a IP-free secretion vector, works in HEC, and I think it works in CHO as well. On top of that, we do actual research. So we use the antibodies that we produce to study a lot of different receptors. We're mainly focused in the neuroscience space right now. So looking at the synaptic cleft, looking at, we've also studied the opioid receptor with several antibodies that bind to it. And the focus of my talk today is going to be on education. So the antibody platform at IPI, this is a schematic of it. We start by producing our proteins of interest in mammalian cells. We purify them to crystallography grade purity. We put them into our yeast display platform where we are able to identify the Fab sequences that bind to the protein of interest. We take the yeast from the sorting, we perform next generation sequencing, and then we use machine learning based tools to prioritize which antibodies we should be making. So we'll eliminate ones that are most likely to be polyreactive. We've got thousands and thousands of antibodies at this point that we know are polyreactive, and so it helps us predict which antibodies we should make and which ones we shouldn't. We take those sequences forward, we produce the antibodies, and then we do a variety of biophysical characterization on them, as well as application testing. So running ELISAs, running SPR, BLI. And then we work with the community to validate that these antibodies are actually useful. So we want to make sure that they work in knockout tissue or they work in cell lines that you maybe knocked down. This whole platform is underpinned by automation. And over the last 3 years, we've really accelerated it by adding a lot of automation equipment. And if you're interested, I've got a poster, P4. We can chat about it after. So why do we care about automation at IPI or education at IPI? So we're really wanting to make sure that the next generation of protein scientists have the tools and the skill sets to move biomedical research forward. We focus on 3 main areas: hands-on training, knowledge exchange, and outreach. So this is a question that I like to ask automation engineers when I meet them. How did you start in lab automation? Josh, Fred, thank you so much. for your talks today. I learned a lot about how you started in automation. I saw some interesting devices that I want to chat with you about. I usually hear from people, someone gave me a chance, someone invited me to try, someone trusted me with an instrument. I don't usually hear answers like, I have a degree in lab automation. I took an automated science course at university. I attended a short course in lab automation. And I think a lot of the people in this room know that many career paths can lead to an automation career. Whether you're on the hardware track as a mechanical engineer, software track as a software developer, or maybe you started as a strain engineer or a protein engineer and you ended up in automation. But career paths don't often start in lab automation. And as a hiring manager, that becomes abundantly clear when I post a position for a lab automation engineer. I'll get a stack of resumes that are, comprise excellent scientists, software and hardware engineers. But it's extremely challenging to find someone who has the skill set, either on the hardware side or the software tools that we use every single day in the lab. It's, can be very challenging, even in the Boston area, to find someone. So if we look at the field more broadly, it's expected to grow by about 10% every year for almost the next decade. Yet there's only a handful of academic courses in North America that are offering any sort of training in lab automation. And so if we want the field to continue to grow, we need to make sure that we're investing in the workforce and providing the training to support that growth. And we're going to need more traditional routes to get people into lab automation. It can't just be chance opportunities. We all ended up here by chance, but if we had more explicit pathways, maybe this room would be 10 times larger. We think about scientists. They're trained to think, to understand the biological context. They're going to pipette by hand, and they're going to design experiments for the bench. They're not trained how to design workflows on an integrated system, debug hardware and software issues, or automate liquid handling. And so what I wanted to do is start to address that skills gap, especially for people coming out of college, and put together a course in automated liquid handling. And so last year, I wrote a proposal to our education team for automated liquid handling in January. Sent it off to them. They came back to me in February and they said, we don't really know what this is. We need more information. Can you tell us what it is? So we talked about it and they said, well, we've been talking to Dr. Deegan at the Emmanuel College. She's the Dean of Science and Health. Let's send it to him. Let's see what he thinks about it. So about a month went by and we got an email back that said, I shared this with our faculty. We love the idea. Can you run the course in July? Now, initially, when I wrote the proposal, I thought, I'd have like a year to develop the curriculum, put the course together. I had 3 months. So I had to do it a little bit faster than I was expecting, but we said we would do it, and we delivered the course last July. This is the course that we put together. So it's a 2-day course designed for students who have basic wet lab knowledge. We kind of just assume that they have, they've used a pipetter before, but we assume they have no automation experience and no coding experience at all. Our learning objectives were that they would understand the principles of automated liquid handling, automated pipetting techniques. We want them to be able to develop a new method on a liquid handler, write and modify Python scripts to run the liquid handler, be comfortable setting up, running, optimizing methods on the liquid handler, and then exploring career pathways in lab automation. So on the first day, we give the students a 1-hour lecture on pipetting techniques and an introduction to lab automation equipment. We then spend the morning building a copper sulfate assay method. We use the protocol designer from OpenTrons. It's a very easy way to teach people because they can just go to a web browser, pull up the software. They don't have to install anything. Makes it very, very straightforward. And we walk them through building that assay. So you have to perform a serial dilution. This is copper sulfate. It's nice and blue. It's great colors for them to actually see the liquid handling, make sure that it's actually mixing properly. That afternoon, the students actually run the method that they wrote. So they're building the method along with me. They then load that method and they actually do a dry run on the OT2. And then they do a wet run and they optimize the liquid handling in the afternoon, getting a nice standard curve and determining the concentration of an unknown sample. On day 2, we give them 1 hour lecture on Python. And what we do is we provide them with a copper assay script and we walk them through each of the steps of the method and we connect it back to how they built the method in the GUI. So this is how you aspirate it in the GUI. This is how you do it in the Python script. They spend the morning converting the copper sulfate assay into a Bradford assay. It's a protein quantification assay, uses another dye that turns blue when it interacts with protein. There's a few additional steps they have to add. All of the students are very intimidated. The students we've worked with, they've never coded before, but we really, we break it down for them. We support them through the workflow, and they come out the other side having written Python code, and then they spend the afternoon optimizing that method on the liquid handler, getting a standard curve, and determining the concentration of another unknown protein sample. We bring the students back to the institute. All of this coursework is done actually at Emmanuel College. They have a great training lab, so it's really easy to work in. We bring them back to the institute and we give them a tour of our lab space. We show them our integrated systems and all the liquid handlers we have. We do some demos, talk about the different techniques and methods that we run on these instruments. And then we host a career panel. So we talk to, we invite automation engineers from the Boston area to our lab to meet with the students. They discuss their career pathway. They discuss, what they're looking for in entry-level roles. And then the students are able to ask questions about lab automation and anything that they're curious about. I really appreciate all the panelists who come to these events. They volunteer their time. We've had some great panelists, including Sam Muller from Lila, some people from Generate Bio, Takeda, Kalpesh Gupta, who's very active in the automation community. He's the director at Moderna. I really can't thank them enough for really making a difference in the course and spending time and talking with the students and staying late and answering all the questions. We are trying to be rigorous with the course. So we're hoping that we can apply for grants to help support the course long-term. To do that, we do a pre and a post test so that we can show learning gains from the students. We do see significant learning gains. And really what we find is that the students feel prepared to walk into a lab and work with liquid handlers. They're not necessarily going to be, writing methods on a Hamilton on day 1, but at least they've seen these devices, they're familiar with them, and they have confidence to walk into a lab and say, can you show me how to, run a Hamilton? And really what we find is it's really helping students envision what a career in lab automation would be. 2 days ago, they had no idea anyone in this room existed. Now they understand what we do, what the kind of work is going to be like. And they leave really excited about lab automation. And so this is at the end of the course with Emmanuel. We tried to figure out what are we going to do next? And that's where we kind of kicked off this year of pilots. So we have this course. What's the audience? Is it just undergraduate students? Could it be for people who maybe don't have a college degree? Could it be for a more professional audience? So we decided we were just going to reach out to the community, see who has interest in this program, and we'd run a pilot with them. So one of the first organizations we spoke with was the Massachusetts Life Science Center. They support organizations who help train people to get into the life sciences. They put us in touch with 3 organizations that had received their Pathmaker grants. So HMS Med Science, Just to Start, and Northeastern University. And we've run pilots with HMS and Just to Start, and we're going to be running one in the fall with Northeastern. HMS and Just to Start have loved it. We've already run a second round of the course with HMS. We're planning another one with Just to Start. And then we're also going to be continuing our partnership with Emmanuel College, running the course in July again. We are also thought maybe it would be applicable to professional audiences. There's a lot of scientists out there who end up in automation. Maybe we can attract some of those curious minds to our event. I'm happy to say that we ran the course at PEG Boston back in May. We sold out the room. We had 24 slots. We had 24 attendees, and it was heavily attended by senior scientists and principal scientists. A lot of curious people wanting to learn more about liquid handling and automation generally. We asked them why they attended. I heard very similar stories. They've got a liquid handler in their lab, it's collecting dust. They wanted a little training so that they would feel confident dusting it off. Or, their team works with automated liquid handlers, but they've never had any formal training and they just wanted to understand how the devices work. We had a couple of people say that they were, interested in purchasing a liquid handler, but they didn't know who to talk to. They didn't know anyone who would be like an objective source of truth. You can talk to salespeople, but they're trying to sell you something. And so they just wanted to educate themselves so that they could be more informed when they're talking to vendors. We're also trying a public course. So this summer, after our course with Emmanuel College, we're going to run a course that's open to the public on July 23rd and 24th. Registration is still open. If you've got anyone who could use some training, let them know if they're willing to come down to the States. I'm really happy that SBT Lab Tech and Nova Biomedical has sponsored the course to help us cover some of the costs. We've been running these things trying to do cost recovery, but we're investing heavily in the course to make these things happen. And I just want to leave you with this student testimonial. So this is a student from Just to Start, Slan Sheridor. She was not part of the Just to Start biotech program. She was actually part of the IT support technician program that Just to Start runs. So this is a video from her. I just want to point out these last two quotes as well. So this was the day I almost changed careers. And so after all of this, I'm happy to reintroduce myself as a lab automation convert, still IT, just upgraded. And I really think that probably speaks to a lot of people in the room, right? You maybe started in the science track or a hardware track. Now you're a lab automation engineer. You still are a scientist, you're just upgraded. Now you know lab automation. I want to, acknowledge the automation team, Eloy, Sophia, Sam, Chris. They worked really hard over the last year. We've run 6 instances of this course in a year's time. Emily and Trisha are the education team at IPI. They've kept the train on the rails the whole time, as we've just said yes and yes and yes to everything. The whole antibody platform at IPI, and Tim Springer and Chaplin Liu, who founded the Institute and support the work that we do. And thank you, and I'd happily take any questions. Any questions, comments?
All right. Thanks, Curtis. Yeah, I think this is very important what you're doing. Just like a lot of people, we all learn automation, on our own, right? And it's true. There's no program. And when you think about AI, now there is program at universities, but lab automation, there's still nothing, which is kind of surprising because the pharma has been doing this for a long time. But anyway, My question is, the first one is the training program. Where is it located?
We do it in Boston.
So the one that's coming up in July?
Yes. Yep.
Okay. And then what I'm curious to know is, you have all the people that are just coming in just to learn more, and then, okay, I know what it is. Gonna go back and then, right? Or
Mhm.
some individual that.
I don't have those stats. I can tell you that we've hired 2 people out of the program that took the course. So we are hiring those individuals, right? So that's the nice thing about running the program. I can find the best of the best and bring them into my lab. Um, I- the workforce development programs that we've been working with, they do a really good job of keeping track of their students. So I think we will start to get some of that data from them to see where those students end up. One of the things that I want to work on over the next year is really curating that talent pipeline to help those students get entry-level positions at biotech and pharma in the area. A lot of the students who are taking this course don't have a college degree, and the Pathmaker program is really designed to help those students, get the basic lab skills to get that first lab technician job. And so I think this is one way to differentiate their resumes when they're applying for jobs. They're going to end up in a lab where they're going to see and work alongside liquid handlers and automation equipment. And it's a gap that, no one else is teaching that information to their students.
Great. Any other further questions or comments? I think this is one of the great things really to come out. And I think as the LRIG board also, we've kind of identified this across the country as a huge hole, right? I mean, like you say, you learn it on your own, you wing it, you crash a few robots, fix them, and try to do it again. And a lot of that, we're fortunate now that we have some experienced people teaching these people slowly, right? But at the same time, it'd be great if there was some kind of course. And within LRIG, we're trying to see, can we get manufacturers, the industry people, like, is there a willingness to develop an automation type course? And I'm not saying specific, but generally, walk into labs, a 2-day course is great. I think, Chris has done a wonderful job here, or Curtis, sorry, has done a wonderful job here, putting this together, right? But you can see that, you could hold a week-long summer course with somebody and really get them up to a fair amount of speed where they'd be very comfortable walking into a situation like that. So just really let everybody think about that, from the LRIG Canada side, we'd like to see something develop more, and we're looking for volunteers potentially to help us out and drive that program. As a board, we're doing as much as we can to run this meeting once a year. We don't have the bandwidth to do everything. So we'd really like to listen and hear from people. And maybe we can bring Curtis up sometime and, start a pilot program and start walking across the country here, to go on the backbone of his 2-day course, extend that maybe out. And I'm sure that we could exchange resources and try to make this something, nationwide, countrywide. Sorry, another comment here?
Yeah, perfect. Yes.
Hi, I'm Christine. My question or my idea was like, I would be personally very interested in having like online courses that go, let's say, in best practices of programming or something like this. And I think that would be like accessible, like maybe US wide and Canada wide, for instance. Do you have any plans to do that?
We do. So we're working on some videos that we're going to be releasing, hopefully in the fall. One of the other challenges I think in the field is that so much of the educational material is behind vendor paywalls. You've got to get the instrument, you've got to spend the $5,000, $10,000, $15,000 for the training program. So we're trying to figure out what we can make accessible and available to the community. From just, how do you use this feature, or how do you solve, this particular problem in your methods? So there's definitely more coming. We're also thinking about like a longer course where it wouldn't just be on automated liquid handling, but we'd be looking at like using a whole integrated system and spending a week on it. And, take it apart, put it back together. Let's do integrations, maybe like, let's write some drivers. I think right now we could do whatever we want. And so it's just trying to prioritize. And this is not our day job. This is like extra that we do. Like it's a hobby basically for us. We do as much as we can. And I'm hoping that it'll continue to grow.
Great initiative, great talk. I was not really a question, just a comment. I was at the University of Toronto for the past 10 years, and they have a new big project funded by CFRF, I think, big grants, called the Acceleration Consortium. And they're all about automation and AI. And I believe they have training programs as well. So that would be perhaps an opportunity to reach out to them. Perhaps they are offering already some.
Yeah, this meeting is going to Toronto next year, and we've had contact with that group previously, and I think we're actually going to get hosted by them next year. So that would be great interaction. They are not as much bench scientist side automation. I know they're more the other automation, but automation's automation. So there's lots of cool things you can learn both ways. So yeah, that's a good point. We'll definitely look at potentially piggybacking onto some of what they have and what they're learning. But once again, it's really the science lab automation. That's what LRIG's here for, is really to try to get scientists to become more, let's say, automation friendly as to moving forward and more comfortable with it. That helps everybody. It helps the vendor sell more equipment. It helps science go faster, et cetera, et cetera. So I think we should all be, advocates for something like this. So thanks a lot, Curtis, for your talk. I think it's going to cause a lot of conversations to happen, hopefully, and
I hope so.
we'll come back around to you. So that kind of concludes SLAS. Sorry, not SLAS, LRIG. Going back and forth for meetings here. LRIG 2025. What we're looking for, if you look at the day that we've had, we brought together a whole bunch of vendors. Hopefully you've got some snapshots of that. Hopefully the vendors got something out of it, got some people interested in their products. And that's the big thing about this is we're all volunteers just trying to put this together. We're always looking for more volunteers from various areas. Please, if you have some time on your hands, nominate yourself, come up and talk to us and say, hey, I'd like to help out, because it's kind of tough on the 5 of us. We got 5 of us and a couple other volunteers, and we need more. If we have more volunteers, we can accelerate some things like these training programs. We need a little more help on the web side of things. We'd like to do some webinar type things, which we think would be useful in automation. And remember, these are all focus specific automation. So they're things that you are hopefully interested in. It's not just another, webinar where you actually get 2 minutes of goodness out of an hour of listening. So if you are, please help us out. We thank you very much for coming out. Thank you very much to the vendors for supporting us in this year. And we hope to see some of you in Toronto. Thanks a lot.